Hello, I'm Scherer from Saarland University, and I would like to present to you my poster entitled Quantitative Comparison of Within-Sample Heterogeneity Scores for DNA methylation data. In this very collaborative project, me and my co-workers, we worked on a level of heterogeneity that is barely addressed in epigenomic studies, namely within-sample heterogeneity. In a classical epigenome-wide association study, or EWAS, a group of healthy individuals is compared to a group of affected individuals, let's say a group of cancer patients. We then determine so-called differentially methylated regions, which are significantly different between the two groups. Within one of these phenotypic groups, you can still have some degree of heterogeneity that is mainly driven by cellular composition. And you can address for this heterogeneity within group heterogeneity using deconvolution approaches. In this project, we worked on within sample heterogeneity, which is at a given genomic location, the variation in the sequencing reads that we observe within a single sample. And this WSH within sample heterogeneity can be driven by multiple sources of variation. Most notably, cell type heterogeneity contributes for each of the cell types a distinct DNA methylation pattern to the sequencing reads that we observe. And in these sequencing reads, you can then determine epi-alleles, which are configurations of CpG methylation states in 4 CpG windows. You can also define the concept of concordance or discordance, where a concordant read pair is a read pair where all CpG methylation states of the CpGs that are shared between the two reads are the same, and discordant otherwise. The second source of WSH is cellular contamination. So imagine you have a set of purified cell types which have been purified, for instance, using FACS. But this cannot work perfectly, and in the end you will have a cell type that contributes a distinct methylation patterns to your purified cellular population. Then you have allele-specific methylation, where only one of the alleles is methylated, which also contributes to these heterogeneous reads. And last, there's DNA methylation erosion. So if you have a high level of DNA methylation during multiple rounds of cellular division, this DNA methylation can be lost, since DNMT1, which is responsible for copying the DNA methylation state to the daughter strands, is not working perfectly and makes errors. There are genome-wide scores available to quantify WSH. And we first of all benchmark them by defining them conceptually into two groups, namely into intermolecule scores, which comprise epipolymorphism entropy, and our two scores that we present, the fraction of discordant read pairs, FDRP, and its quantitative extension Q, FDRP. And these intermolecule scores describe the variations of the sequencing reads, while intramolecule scores rather describe the heterogeneity in a given sequencing read, and these scores include the proportion of discordant reads, PDR, and methylation haplotype, MHL. We then used simulated data and simulated these different sources of WSH, cell type heterogeneity, cellular contamination, allele-specific methylation, DNA methylation erosion, and we also simulated a data set where we don't have heterogeneity, but rather a switch in the DNA methylation state, as you would expect them at the border of active regulatory elements. We then define so-called truly heterogeneous regions, THRs, which show an elevated level of heterogeneity, and then use ROC curves to determine whether the scores reliably differentiate between the THR and the background. We found that FDRP, QFDRP, epipolymorphism, and entropy reliably detect most of these THRs in the 1000 simulation experiments that we conducted. In contrast to that, PDR and MHL do not differentiate between the THR and the background. We also found that especially QFDRP and MHL are robust with respect to technical biases, including read coverage and read length. We then applied QFDRP to a childhood bone cancer dataset, Ewing sarcoma, and found that QFDRP can be used as a reliable estimate of tumor purity. We then also applied QFDRP to define differentially heterogeneous genes, and these differentially heterogeneous genes were enriched for Ewing sarcoma specific features. Last, we then provide recommendation of and guidelines for the different scores. So for instance, we found that epipolymorphism and entropy reliably detect the heterogeneity in our simulation experiments, while they do not provide a CBGY score. In contrast to that, the scores that we present, FDRP and QFDRP, provide a CBGY WSH score, which make them a perfect candidate to associate CBGY's WSH scores with CBGY's DNA methylation levels. In addition to that, QFDRP is also very robust with respect to technical setup. So if you are interested more into this topic, please come visit me virtually at my poster. We also have an associated publication available in NAR and an R package available from GitHub. Thank you for your attention.